Hello, everyone, and welcome to phyloseminar.org. The current theme is branch-specific diversification inference, by which I mean learning about the diversification process via phylogenetics. If you want to ask a question, you can either tweet at phyloseminar or type your question in the live chat box to the right of the video on YouTube. Today's speaker is Ben Good. Ben develops methods to infer fitness and predict evolutionary outcomes. He did his PhD with Michael Desai at Harvard and then was a Miller Fellow at UC Berkeley with Oscar Holacek. And now he has just taken a job at Stanford in applied physics. Welcome, Ben, and thanks for participating.
Awesome. Well, thanks, Eric, for that kind introduction. It's a real honor to speak here at my first Philo seminar, and I'm really excited to tell you about some recent work we've been doing to try to understand the population genetics of adaptation and ecological diversification on substitutable resources in resource competition models. And so the broader theme of this Philo seminar series is the interaction between fitness and trees. And so I just thought I'd start today's talk by quickly reviewing what I think we understand really well about this process and where today's talk fits in. So as usual, the trees that we understand the best are the ones that arise in the absence of natural selection, where all of the individuals in a population, any new mutations that arise, are all selectively neutral with respect to each other. Now, the genealogies of a present-day sample in this scenario are described by Kingman's coalescent, where every pair of lineages has a probability 1 over n of coalescing into a common ancestor as we sort of march back in time. And forward in time, this corresponds to mutation trajectories, sort of like the one I've illustrated here, which can be described by the familiar Wright-Fisher diffusion process that models genetic drift. So this neutral scenario has the advantage that these forward and backward processes are often exactly solvable. And one of the key findings is that the typical timescales of fixation of mutations or coalescence backward in time end up being proportional to the population size n. And so this means that even for relatively moderately sized populations of 100,000 or 1 million individuals, these neutral processes can be extremely slow, leaving plenty of time for additional diversity to build up in the population through new mutations, which we can model by allowing mutations to accumulate on the branches of these trees. So the effects of natural selection are easiest to illustrate if we think about the classic picture of a single selective sweep, where we imagine that some new beneficial mutation arises in a single individual in the population and confers some growth rate advantage plus S to its descendants. Now, if this mutation is lucky enough to survive drift, it'll start to sweep through the population on a timescale that now only grows logarithmically in the population size n. However, if we look backward in time for large populations, most of the lineages from a present-day sample won't actually have an appreciable chance of coalescing until the first 1 over s or so generations in the time since the mutation first arose. At this point, they're going to have to coalesce extremely rapidly, since they all eventually have to pass through this single individual bottleneck when the mutation first appeared. And so this means that compared to the neutral scenario on the previous slide, the sort of zeroth order effect of selective sweeps is to tend to make genealogies look shorter and more star-like. And so the key empirical parameter then just becomes the typical time since the last selective sweep actually occurred in the population. Now, this picture starts to become a little more complicated when beneficial mutations are more common, and this typical time between sweeps starts to become comparable to the sweep time itself. Now, in this case, multiple beneficial mutations can be sweeping through the population at the same time. And when this happens, natural selection can't always act on these mutations individually. When recombination is limited, it can only act on combinations of mutations that happen to be inherited together on the same physical chromosome. And so what this means is that mutations have to actually compete with each other in the population through a process known as clonal interference. And sometimes, I've drawn the cartoon here, multiple beneficial mutations can actually be required before a lineage is able to completely sweep in the population. Now, this sort of scenario has actually been anticipated since the really early days of population genetics. So little cartoons like the ones I've drawn over here on the left can actually be traced back to a paper by Herman Muller back from 1932. But the genealogies that are produced by this process have only been understood relatively recently, mostly by studying toy models of really long asexual genomes with large numbers of weakly selected sites, sort of like this little coupon collecting model of a genome that I've illustrated down here. So the genealogies produced by these simple models end up having a relatively universal genealogical structure characterized by skewed branching and these multiple merger-like events. And if you're interested in learning more about these, you can go back to Richard Nayer's excellent Philo seminar from a few weeks ago. For our purposes, it's enough to know that Since this effect depends on the competition between multiple simultaneously segregating mutations, we expect it to be relevant in a regime where multiple beneficial mutations are introduced in the population every generation. Or in other words, when the product of the population size n, the per site mutation rate mu, and the sort of total target size for beneficial mutations is greater than or equal to 1. And so this means we expect these effects to be particularly relevant in microbial and sort of microbe-like populations, including pathogens like influenza and HIV, populations of somatic cells in the body like immune repertoires, and importantly for us, laboratory evolution experiments in model microorganisms. And with modern genomic data, it's now possible to actually go in and measure these population genetic signatures of clonal interference directly. However, when we do so, it's also starting to become clear that these molecular data show that a fundamentally different kind of selection can also be important in these microbial systems. And that's what I want to talk to you about today. So this is easiest to illustrate with an example. So I want to take a quick detour and tell you about some molecular data that we generated from Rich Lenski's long-term evolution experiment in E. coli a few years ago. And so this is one of the simplest laboratory experiments you could imagine. 12 independent populations of E. coli have been serially propagated in glucose-limited media every day for about a little more than 30 years now, and a little over 70,000 E. coli generations. And most importantly, Rich and his team have been careful to preserve frozen whole population samples every 500 generations so that people can go back in and interrogate these populations later. Now, ideally, what we wanted to measure was these sort of underlying Muller plots, essentially equivalent to a time-resolved phylogeny for each one of the 12 experimental populations in the LT. But at the moment, this isn't still quite feasible to do with traditional isolate-based sequencing. So instead, what we wanted to do was actually just measure the population frequency trajectories of all of these individual mutations within the population, or at least those that reached observable frequencies. As you can sort of see, this throws away a lot of information about genetic linkage, which mutations are actually present in the same cells. But it ends up being much more cost-effective for dense temporal data of the kind that we have in this experiment. And actually, in many cases, you can actually recover a lot of the linkage information by thinking carefully about the joint dynamics of these mutation trajectories across the different mutations. Just to give you an example here, we see that these A and C mutations eventually took over the population together. That means at the end, they must be present in the same cells. They occurred at 2 different times. And so the only way for this to happen was if the C mutation actually occurred as a nested mutation in the background of A here. Conversely, the B mutation is always traveling in opposing directions to both A and C. And this suggests that it was actually that arose on a separate genetic background. And so in this way, we see that the only way we can really self-consistently account for these mutation trajectories over here on the right is through the Muller plot or time-resolved phylogeny cartoon that we drew on the left. So I feel a little bit bad in the phylogenetics seminar sort of throwing away trees so early in the talk, but I hope I've convinced you that these mutation trajectories actually contain a lot of the same information. Now, the advantage of focusing on these mutation trajectories is that we can actually measure them with a metagenomic approach, where we grind up the entire collection of genomes that are present in a given frozen sample and then sequence them together in a big pool of short 150 base pair long fragments. The idea is then if we align these short fragments against the E. coli reference genome, we can use these data to obtain a noisy estimate of the frequency of a particular mutation in a particular population at a particular time. And so here in this example, I'm just showing you there must have been a mutation from a T to an A at this particular site in the E. coli genome. And because it's present in 3 out of the 5 sequencing reads that line up to this site, we infer that it must be present at about 60% frequency in this particular population at this particular time point, perhaps with some Poisson counting noise. The idea then is that we can string together different samples from the same population together, and then use these estimates to trace out the population frequency trajectory of each mutation over time. So that's the basic idea. So here's an example of what these data actually look like. This is one particular missense mutation that occurred in the REC-O gene. It arose and eventually took over in one of the 12 replicate populations, which goes by the name ERA-6. This is what it looks like for one mutation. This is what it looks like when we plot all the mutations from the same population together on the same plot. So each line here represents a unique mutation that occurred at a different site in the E. coli genome. So these figures look relatively busy. A lot of this busyness is being produced by clonal interference and the fact that there's just a large number of beneficial mutations that are accumulating in this experiment over time, even after tens of thousands of generations in the same external environment. But if we keep looking at these mutation trajectories, we also start to notice that there's some sort of weird stuff going on from a population genetic perspective. So this is easiest to see if I go ahead and split the mutations into 3 groups for you. So here I'm showing you the same population in each of these 3 panels. I've just gone ahead and colored the different mutations differently in the 3 panels according to 3 different criteria. And so what this partitioning is supposed to show you is that in the early days of the experiment, mutations were appearing and taking over the population as we expect, perhaps with some clonal interference. But after a while, mutations stopped completely taking over the population entirely, and instead seemed to congregate into these 2 intermediate frequency clusters of mutations, or I'll call them clades for today's talk, that actually coexist with each other for about 40,000 generations. Now, importantly, it's not that evolution stops entirely once these 2 clades arise. We sort of zoom in on the mutation trajectories here in these bottom 2 panels. We can see that individual mutations are still rapidly accumulating within these individual clades on timescales that are much shorter than the clade lifetimes themselves. And so this means that this shows up as sort of these deep and really spindly branches in the genealogy of the population, which we can just start to make out from this smaller sample of clonal isolates that were sequenced in a previous study. And so just based on purely qualitative terms, you could imagine that an example like this could arise just as a really long-lived example of clonal interference, where we have 2 asexual lineages competing with each other, and they just happen to get lucky enough and acquire beneficial mutations at roughly the same time in such a way that it prevents any individual lineage from gaining too much ground in the population. However, this is where we can actually lean on the large body theoretical work on clonal interference that's been done in the past 5 to 10 years. A more careful quantitative calculation will show you that the separation of timescales between within clade fixation and the clade lifetimes themselves is actually inconsistent with simple population genetic models of genetic linkage and clonal interference on a long asexual genome. Instead, the data forces to conclude that these clades are stably maintained within the population somehow, presumably due to some form of primitive ecological structure that evolved over the course of the experiment. And now this might sound a little far-fetched, but this is actually known to be the case already in one of the 12 replicate populations. And in this case, the mechanism of coexistence is already known to be mediated by a metabolic cross-feeding interaction that evolved over the course of the experiment. And the idea is that one of the 2 strains evolved the ability to better metabolize acetate, which is actually excreted into the media as a byproduct of overflow metabolism on the primary nutrient, glucose. But it gained this ability at the cost of its ability to actually grow on glucose earlier in the daily dilution cycle. And so these 2 traits combine in such a way that if you take these 2 strains and mix them at different frequencies, they rapidly approach a stable, what I'll call ecological equilibrium frequency, F star, on a timescale of about 50 generations or so in this particular case. This is a form of frequency-dependent selection, and it can't be captured by any constant growth advantage or disadvantage of the type that we've been talking about so far, since the sign of the selection pressure actually changes depending on the frequency that particular mutation has. Now, this particular example was actually identified based on colony morphology of the 2 coexisting strains. These molecular data now tell us that this previous example wasn't just an isolated special case, but this long-lived coexistence is actually relatively common across the different replicate populations in the LTE. So here, these purple and pink lines are showing you the inferred clade frequencies, similar to these black lines over here on the left, for each of the 12 replicate populations in the LTE. And so we see that we have some evidence for long-term coexistence in about 9 of the 12 replicates. Perhaps more interestingly, these data also show that although these ecological interactions seem to be stable on these relatively short 50 generation timescales, they're only quasi-stable when we look out across evolutionary time. So if we follow these lines here, we see that the relative frequencies of the clades can shift by as much as 10-fold at different points in the experiment. And the inference is that this happens whenever one or the other of the 2 clades acquires some special mutation. And so this leads to some relatively high variability across the different replicate populations in the experiment, both in the state of the ecosystem at the final time point, as well as in their temporal trajectories over time. So just to give you an example, this population we've been talking about so far, era -6, the 2 clades arose and coexisted with each other throughout the end of our sampling period. But in the population immediately below it, called era +1, coexistence arose at roughly the same time, and the two clades coexisted with each other for roughly the same order of magnitude number of generations. But eventually, this purple clade was able to rise to dominance in the population and essentially take over. And the idea is, since these different populations are maintained in the same external conditions, these data are telling us that stochastic evolutionary forces must be playing a key role in shaping the structure of these really primitive microbial communities. And so these coupled ecological and evolutionary processes aren't just unique to Rich Lenski's experiment. In fact, with increasing molecular data, we can now observe similar processes of ecological diversification occurring in a lot of other laboratory systems as well. Moreover, if we look outside the lab, there's, of course, long-standing evidence for ecological substructure in many natural bacterial populations, where these two clades would go by the name of ecotypes. There's also some evidence for ecological diversification in influenza B virus, which actually split into two antigenically distinct clades back in the 1970s that have been coexisting with each other in the population ever since. We also expect some similar dynamics to arise during the competition for distinct antigenic stimuli in simple models of immune repertoire dynamics. So these different examples are telling us that ecological diversification can be a really important and potentially widespread factor in microbial evolution. But the population genetics of this process are still somewhat fuzzy. And so this leads to some open questions at the population genetic level, even in these really simple experimental systems like the Lenski lines. So for example, what could explain the relatively common emergence of coexistence across these different replicate populations while allowing for the relatively varied outcomes from replicate to replicate? Do we have to posit some separate biological mechanisms of coexistence in each flask? Or could this reflect some more general coupling between the race to accumulate fitness and the current ecological equilibrium frequency? These ecological interactions accelerate evolution in some ways, since now the two clades have to sort of co-evolve against each other. And more generally, what can we really hope to infer about the underlying ecological and evolutionary processes from at least what appears to be some highly quantitative DNA sequence data? Now, I'd argue that a big problem at the moment is that most population genetic models neglect these sorts of ecological interactions and vice versa. And this means that while it's relatively easy to reject a simple model without any ecological interactions, as we just did in the case of the Lenski lines a few slides ago, we often don't really know what to expect in their place. Just to give you an example, should we be surprised by the fluctuations in these clade frequency trajectories that we measured in the Lenski lines or not? At the moment, I would argue we don't really have the theoretical tools to begin to answer a question like that. And to do this, we need to develop some more quantitative theory. And so today I want to tell you about what I think are some promising first steps towards this goal. So in principle, we should be able to incorporate ecological interactions into these population genetic models just by assuming that all fitnesses are actually subject to frequency-dependent selection, where the fitness of a particular genotype now depends not just on the state of its genome itself, but also on the current frequencies of all the other genotypes that are present in the population. Now, even at lowest order, this is a pretty tough thing to model in the general case, since it requires us to somehow measure or at least assume the interaction terms between all possible genotypes in the population. So to get around this problem, I instead want to focus on a really simple empirically motivated toy model, similar to the Lenski lines, in which strains compete for our different substitutable resources in a well-mixed chemostat-like environment that I've drawn here. So by substitutable resources, I want you to think different carbon sources like glucose or acetate. The idea is then we can write down a joint model that keeps track both of the individual frequencies of the different strains or genotypes in the population, which I'll index by this Greek letter mu, as well as the concentrations of the different resources in the chemostat, which I'll denote by this Roman letter I. And this is a special case of a more general and well-studied class of models from the ecological literature known as consumer resource models. And so in our case, we want to assume that the different resources are supplied by the environment at different fixed rates. And I want to let this parameter beta I denote the fraction of biomass that's supplied in the form of resource I. I then want to assume that the different individuals in the population can take up these different resources at different genetically encoded rates. And I want to represent these by what I'll call a resource strategy, where this parameter alpha mu I represents the fraction of effort that strain mu spends to import resources of type I. This is going to be normalized such that alpha mu I sums to 1 when we add up for each strain across the different resources. And finally, I wanted to note the maximum growth rate or total energy budget, if you will, of each strain by this third parameter, x sub mu. And I'll show in a second, this is going to have an interpretation as sort of a general laboratory notion of fitness. Now, at first glance, it looks like I haven't really solved anything. I've just made the problem more complicated by introducing these additional internal degrees of freedom. But actually, in many cases of interest, the dynamics of these resource concentrations are going to end up relaxing on much faster timescales than the dynamics of the genotype frequencies themselves. And so in this limit, we can actually eliminate the resource concentrations entirely and write down a single coarse-grained model that can be written entirely in terms of the strain frequencies themselves. And so in the case of just a single resource, you'll notice that these dynamics just reduce to the familiar Wright-Fisher selection term that we're used to in population genetics. In the limit of multiple resources, it maintains a relatively similar form, except now essentially we have to allow for a separate mean fitness term that's specific to each of the R different resources. And so in some sense, you can think about this model as being the simplest generalization of the Wright-Fisher model to allow for multiple fitness dimensions at the same time. Now, it's relatively straightforward to convince ourselves that a model like this could allow for stable coexistence in the extreme case where every strain specialized on a totally different resource. But it's also known that this model allows for stable coexistence to arise, even in the case where the strains partially overlap in their resource uptake rates. And this is what's going to make the model interesting from our perspective. Now, on its own, this model only really describes the competition between a fixed set of strains that were created back at the beginning of time. And so to allow for evolution, we also want to allow these strains to evolve over time by acquiring heritable mutations that alter their resource uptake rates in different ways. And so today I want to convince you that it's going to be useful to distinguish between two broad classes of mutations. The first, which I'll call resource strategy mutations, are going to alter the resource uptake strategy of an individual. And this is what's ultimately going to allow for ecological diversification and the emergence of stable coexistence from a clonal ancestor. However, I also want to incorporate a key new ingredient inspired by laboratory experiments like the Lenski lines, which is that bacterial genomes are really long and complex. And there's often lots of non-metabolic adaptation going on at the same time that these processes of ecological diversification are occurring. And so by non-metabolic adaptation, I just want you to think simply deleting a function that isn't necessary in the current environment. And these types of mutations are actually relatively common in laboratory experiments like the Lenski lines. So to account for these effects, I also want to consider a second and potentially more numerous class of mutations, which are called pure fitness mutations, which will increase the fitness of an individual, but will otherwise leave its resource strategy intact. So one way to think about this, it's sort of the simplest genome-wide model that allows for the evolution of a few ecological traits, while also accounting for the large number of weakly selected mutations that we know are going to be important in large microbial populations. Now, at least naively, you could imagine that these pure fitness mutations would be a really small or at least ecologically trivial addition to this model, since whenever they occur, they're always favored to take over relative to their parent strain. But actually, computer simulations show that the accumulation of many of these weakly selected mutations can actually have a really dramatic impact on both the ecological structure and the evolutionary dynamics that emerge in these simple models, even in the special case of just two resources. And so here I'm showing you some simulated mutation trajectories, similar to what we measured from the Lenski lines a few slides ago, for 4 different populations, simulated with 4 different sets of parameters. And so each of these populations has exactly the same environmental conditions and the same access to resource strategy mutations, but they differ only in the mutation rates and the fitness effects of the pure fitness mutations that they have access to. And so you can see this leads to a wide range of different behaviors at the population genetic level, ranging from rapid diversification and stasis on long time scales, to unstable but periodically renewed coexistence, to stable coexistence of this red mutation, but the rapid accumulation of fitness mutations within the 2 types, to the complete disruption of stable coexistence altogether. So I think what I find really interesting about these pictures is that the top 3 panels have exactly the same present-day ecological structure as defined by how many different strains are coexisting and how are they eating the different resources. But they have very different genealogical structures if we look backwards in time. And so this suggests that if we can figure out a way of predicting these genealogical structures in this model, it might give us a way to start to distinguish between different scenarios that would otherwise be indistinguishable ecologically. So this raises a natural question, which is, what are the important combinations of parameters or knobs that control the transitions between these different genealogical regimes? So, or in other words, if I gave you some data like this, how could you tell me what parameters I secretly use to run the simulations with? That's what I want to focus on for the rest of the talk. So to get some insight into this question, it's useful to start from the simplest case, where there are no pure fitness differences within the population, and there's a sort of perfect trade-offs across the different resource utilization strategies. This was a scenario that was actually analyzed by Anna Posfai and collaborators in this really great paper in Physical Letters from a year or two ago. And so I want to think about a scenario where the population is initially clonal with some particular resource strategy, alpha 1, and then a new mutation occurs in one of the individuals in this population and changes its resource strategy to some new value, alpha 2. Now, in the case of just two resources, we can actually represent these different resource strategies in this convenient graphical picture that I first read about in Anna's paper. And the idea is that we can arbitrarily pick one of the two resources to focus on, and then plot the resource strategies on this single real axis ranging from 0 to 1. The idea then is that the components in the other resource are going to be fixed by normalization. And we can also actually plot the environmental supply ratio between the two resources on this same plot. And so with a little bit of work, we can actually show that this mutant individual, alpha 2, will experience an effective selection pressure that ends up being proportional both to the difference between the resource strategies of the two types, as well as the difference between the ancestral resource strategy and the environment. This is going to tend to select for mutations that drive you towards the environmental supply ratio beta, although it can certainly allow for overshooting. Now, if this mutation is lucky enough to survive drift, it will eventually start to increase in frequency in the population. At this point, one of two things can happen. Either it will take over the population entirely in the form of a selective sweep, or it will come to coexist with its parent strain at some stable intermediate frequency that we'll denote by F star. And so Anna and her collaborators actually worked out the conditions for each of these two scenarios. And it turns out that the selective sweep scenario will happen in the case where the mutant resource strategy lies between the ancestral resource strategy and the environment, while stable coexistence will happen when the mutant and the ancestor lie on either side of the environmental supply ratio. Now, it's relatively easy to work out what the equilibrium frequencies are in this case. And when you do so, you find that actually the state of the ecosystem sets itself up in such a way that the population average uptake rate, which I'll denote by alpha bar, exactly matches the environmental supply ratio. So in this sense, we can think about this model as sort of selecting for the ecosystem as a whole to match the environment. In the case of coexistence, this simply occurs when the best way to do this is to keep both strains around at some intermediate frequency. Now, as time goes on, eventually genetic drift will introduce some number fluctuations that will start to perturb these stable coexistence states. And although these can be relatively large in the short term, they quickly average out on longer timescales in such a way that there's very, very weak selection pressure for additional strategy mutations once this stable coexistence has arisen. So at this point, we say that the ecosystem has saturated, since these ecological selection pressures have now vanished. And the balance between the number of coexisting strains and the number of resources matches the traditional intuition that we're used to from ecological coexistence theory. So what we'd like to know now is, how do fitness mutations change this picture? So now I want to think about a scenario where the population has already diversified, and there's two clades coexisting at some equilibrium frequency, F star. Now we want to consider a new mutation which occurs in one of the two clade backgrounds and provides a pure fitness benefit plus S relative to its parent. Now, if this mutation survives drift, it will start to increase in frequency in the population and will now generally lead to a shift in the current ecological equilibrium frequency between the two clades. So in a relatively broad range of parameters, we can actually work out what the shift is. And it turns out to be proportional to the fitness difference between the two types, where this constant proportionality, X star, gives a measure of sort of the evolutionary robustness of this community to the accumulation of new fitness mutations. In this particular model, it turns out that this evolutionary robustness, X star, ends up being proportional to the square of the difference between the two resource uptake rates, which actually coincides with the traditional measure of niche overlap that's been defined in these resource competition models. This means that all else being equal, two clades that have very similar resource strategies are going to be more sensitive to the accumulation of pure fitness mutations in one type or the other, while two clades that totally specialize on different resources will actually be immune to their effects. So the idea then is if this a new fitness mutation, if the fitness benefit S is on the same order as X star, then the shift in the equilibrium frequency will be of order 1. And there's a really large chance that this red clade will end up sweeping to fixation in the entire population and driving the white clade to extinction. On the other hand, if S is much smaller than X star, then the two clades will continue to coexist in the population. And now there's an opportunity for additional fitness mutations to arise in one clade background or the other. So the idea is that in this regime, we can model the accumulated fitness difference between the two clades by an effective diffusion process. And in the weak mutation limit, this effective diffusion process actually takes the following simple form, which I've written down here below. And so the idea is that the bias in the rate of adaptation here arises from the fact that larger clades are going to be proportionally more likely to produce the next successful fitness mutation, while the noise reflects the fact that the fitness mutations will otherwise be arising randomly in the different individuals in this population. And because the total fitness difference ends up being proportional to the shift in the frequency, this induces a corresponding effective diffusion process for the current ecological equilibrium frequency itself. This is sort of the theoretical analog to the fluctuations in the clade frequency trajectories that we measured in the Linsky lines a few slides ago. Now, as time goes on, eventually one of these two clades will acquire a sufficiently large number of fitness mutations that the total fitness difference, delta X, will be of order X star. And at this point, the stable coexistence is going to collapse as the red clade is actually driven to extinction. Yeah, what's up?
Can you just go back and show that comparison again? I mean, I'm just sort of curious, just not so far, but just, yes, that. So let's see, at this point, do we know enough about N and S to be able to say, are these wiggles sort of accumulation of new mutations, or are they just this square root of N level variations that we should be seeing as noise?
I see. Yeah, so that's a great question. I would say my expectation at the moment is that the really big ones are going to be caused by primarily new mutations. There are some sort of wiggles here, and in each case, what's useful is to think about how many mutations are actually contributing to these different trajectories. So a lot of the noise is going to arise just due to sequencing, sampling noise when we do the sequencing. And so in the example I showed you from a few slides ago, these different clades each had maybe, I don't know, 50 or 60 mutations in them. And so these colored lines here represent an average over lots of different mutations. And so we expect this sort of sequencing noise to average out a little bit. In the case of the Linsky lines, the effective population size is actually quite large. So it's on the order of 30 million or so. And so we actually expect that genetic drift on its own is very, very weak effect in these populations. The main difference between these what we measured over here and the sort of theoretical predictions that I'm showing you on the left are that, at least at the moment, these theoretical predictions are totally in the weak mutation limit, where these pure fitness mutations are arising one by one, and we don't have to think about clonal interference between the different fitness mutations. So that's obviously going to be violated horribly in the limit where the effective population size is 30 million or so. And so one of the sort of future directions that we really need to do to actually make contact between the theory and the data is to understand these effects. If you actually take this weak mutation limit really seriously, you can actually convince yourself that the fluctuations in these trajectories here on the right are actually too big. They would never really be produced in a model like this, which kind of runs away as soon as one of the clades gets sufficiently big compared to the other. So that's a really good question.
Thank you.
Okay. Right. So when one of the two clades eventually acquires a sufficient number of fitness mutations, the other will be driven to extinction. And so at this point, there's now going to be a new selection pressure for the winning clade to rediversify according to the same processes we described on the previous slide. Right, so there's going to be a timescale tau collapse that we can predict from these effective diffusion processes below, and then a corresponding timescale tau diversify, which depends on the ecological selection pressures from the previous slide. And so the idea then is that the competition between these two timescales is going to lead to a sort of a dynamic steady state of diversification-selection balance. where if we look at the population over time, it can end up spending long periods of time pinned at values below the maximum capacity permitted by this simple ecological model. And in simple scenarios like this weak mutation limit down here, we can actually work out some scaling relations for how diversification-selection balance is going to depend on the underlying parameters, including the mutation rates for fitness mutations versus strategy mutations, and this evolutionary robustness parameter, X star, which depends on the resource uptake strategies of the two types.
Sorry to stop you again, but I'm a little bit confused about the arrival of new strategies, new alphas.
Perfect.
Okay.
Yeah. I'll keep going.
Okay, so but is this a new part of the model, or?
Yeah, so far, I guess up until this point, we've just thought about, the population has already diversified, and now there's no more strategy mutations, and now we're just thinking about the effects of new fitness mutations occurring one by one.
But in the next, not the next bit, yeah, that right there. Exactly. Well, what is it that caused, so is alpha-2 hanging around ready to be injected into the population?
Yes, the idea is we're thinking about a scenario where there's access both to fitness mutations and to strategy mutations. And so this
I see. So we just flip right to another pre-existing strategy mutation or strategy.
Or a new one that appeared right here, right? If it appeared once in the population, it must be possible for this background to produce it again.
Yes.
It's not really beneficial throughout this whole period, but it'll finally be beneficial again.
Okay.
Right?
Sorry, I get it now. Thanks.
Yeah, so you're hitting on a key factor that I've left out here, which is what if additional strategy mutations actually occurred as this process is going on, right? So something more like this. So before the extinction actually happens, it's always possible that this original clade alpha-1 produces some new strategy mutation. It could be the same as the one that initially established coexistence, alpha-2, or it could actually be something totally different. We sort of have no idea. So it's also useful to consider this scenario here. So if you remembered back from the case where we were considering perfect trade-offs, no fitness mutations, there was just really weak selection pressure for additional strategy mutations after coexistence has been established. And so this might suggest that a scenario like this, we really wouldn't have to consider. But actually, in the case of fitness mutations, you can actually show that the residual fitness differentials between the two types actually lead to an induced ecological selection pressure for new strategy mutations, which actually depends on the fitness difference between the two clades at any given time. And so in the case where we didn't have any fitness mutations, this difference was 0, and that's why we didn't really have to think about new strategy mutations in the case of perfect trade-offs. But now it'll be a little bit different. And so we can work out the various consequences from this equation here. But I actually think it's a little bit easier to understand if we look at this following graphical picture, where we actually plot the new resource strategy of the mutant individual. Sorry, I switched notation. Alpha-3 is alpha prime, and the total fitness of that mutant individual on the y-axis. And we can also stick the two ancestral clades colored in blue here. The mutant will be the little red dot. And so the idea is that a pure strategy mutation that occurs in the fitter of the two clades is going to change its resource uptake strategy, but leave it with the same general fitness as its parent. And so in the case where the mutation occurs in the fitter of the two clades, mutations that actually drive the resource strategy towards the environmental supply ratio, beta, are actually going to be selected for, while mutations that drive the resource strategy in the opposite direction are actually going to be selected against. And then the idea is that depending where this new resource strategy falls relative to its two parents and the environment, this new mutation can either sweep out its parent clade and continue to coexist with the opposite niche. It can actually sweep out the opposite niche and then start to coexist with its parent. And there's actually a narrow region in the middle where you can show that it sweeps out both of the pre-existing clades and rises to dominance on its own in the population. And so it's maybe not so surprising that mutations in the fitter of the two clades will want to try to be as promiscuous as possible in a model like this. What's maybe a little more surprising is that there's an entirely mirrored set of selection pressures for mutations in the less fit clade as well. And so the idea here is that mutations that drive the resource strategy towards the fitter of the two clades were actually going to be selected against, while there's a net positive selection pressure for mutations that drive the resource strategy even further away from the fitter of the two clades. So we can see that these fitness differences are in general going to generate emergent selection pressures within the population, they're going to tend to select for fitter generalists and less fit specialists. So it's really worth pointing out here that this sort of induced fitness landscape is going to be highly dynamic. And so the sign of the selection pressure on a particular mutation can actually shift and even switch signs over time as the relative fitnesses of the two clades change as they accumulate additional fitness mutations within them. So you can imagine possibly testing this prediction using engineered mutations in simple laboratory experiments like the Lenski lines. Okay, so we've seen how it's possible to get a relatively detailed picture of what's going on, at least in the case where we think about pairwise coexistence on two resources. Of course, a major advantage of these resource competition models is that they allow for an arbitrarily large number of strains to actually coexist with each other. We simply keep increasing the number of resources in the chemostat. And so this creates some exciting opportunities to maybe extrapolate from these simple examples of pairwise coexistence that we see in the laboratory to something that's at least starting to look a little bit more like the more complex microbial communities that we see in nature, which can often contain hundreds of coexisting strains and hundreds of different resources. So of course, just simply increasing the number of resources in this model is going to start to make things more and more complex, since now the environmental supply ratios and the different resource uptake strategies have to live in these increasingly higher dimensional simplexes, which are just going to involve a large number of additional parameters. But maybe as a naive physicist, we also might expect that maybe some simplicity will start to emerge again if we start to think about things in the limit of an asymptotically large number of resources. And indeed, some really exciting recent work by Pankaj Mehta and Michael Tikhonov and many others has shown exactly that. That it's actually possible to gain some analytical insight into at least the ecological properties of these models by utilizing techniques originally developed for studying the physics of disordered systems. And so these studies have mostly focused on a process known as community assembly in ecology, which tries to model how a new community sets itself up on a previously vacant environmental patch. And the idea is that we draw some fixed ensemble of colonizing strains, S0, from some global species pool. And then we allow these strains to compete with each other according to the ecological dynamics, these consumer resource models, and ask who survives once this ecosystem has reached its ecological equilibrium state. Now, it turns out that these resource competition models are sufficiently simple that you can actually attack this problem analytically using an approach from physics known as replica theory, where we model the resource uptake rates of the different species and the different other parameters in the model as a form of quench disorder. So one of the key results from these calculations is that if you draw a number of colonizing strains that is proportional to the number of resources, as we let the number of resources grow increasingly bigger, you really quickly reach a state of ecosystem saturation, where the number of surviving species is roughly the same as the number of resources. And this state has some generic statistical properties at the ecosystem level that are relatively preserved across different random realizations of this colonization process. And so one way to think about this emergent predictability is that it arises due to a form of the central limit theorem. And the idea is that in these really large and diverse ecosystems, each one of the resources is going to end up being utilized by a large number of different strains once the community has settled down to its ecological equilibrium. And vice versa as well. Each of the strains that survives in the long term is going to end up doing so by eating a relatively large number of different resources. And so this means that any randomness in the individual uptake rates are going to start to average out and lead to some more predictable behavior for ecosystem level quantities. And this has generated a lot of excitement within the community. It's a way of potentially dealing with the countless unknown parameters that would be present in any real microbial ecosystem. And so this universality from these replica theory approaches suggests that we might actually be able to make some good predictions despite all this biological uncertainty. So given what we've discussed so far, I think it would be extremely interesting to know how these results change if the strains aren't now fixed for all time, but if they're actually allowed to evolve and acquire heritable mutations over time. And this could be potentially really important. So you often read about analogies between random migration events from a species pool and random mutational changes due to acquired new mutations. But there's actually a really important difference between these two processes, which is inheritance. So if you think about it, this community assembly process in some ways is essentially genealogically trivial, in the sense that the genealogical relationships between the different strains that coexist at ecological equilibrium are entirely dictated by the genealogical relationships that you have to pre-program in your species pool. Now, in contrast, if we allow the strains to evolve over time, they will generate some emergent genealogical correlations within the ecosystem just due to evolution. And these genealogical correlations are known to cause some big problems for these existing replica theory approaches. And so in principle, modeling evolution is harder because it requires us to assume a whole bunch of additional parameters about how the different resource strategies can change over time. But as a first pass, we could try to get these effects a little bit by studying a really simple toy model, similar to these existing replica theory approaches, where we allow the strains to just toggle between using or not using each of the different resources at some particular rate, and then assume they can also acquire these pure fitness mutations of a characteristic effect size S at some constant rate ux. Now, if we simply extrapolated from these community assembly results from before, we might imagine that after an infinite amount of time, we expect these ecosystems to eventually reach a saturated state where the number of current species in the chemostat is roughly equal to the total number of resources. And the idea is just that at this point, evolution has now been able to test a large number of randomly generated strains. However, it turns out that depending on the mutation rates and the fitness effects of the pure fitness mutations that these individuals have access to, we find these ecosystems can actually approach an intermediate regime, which is still really diverse, so many more than one species coexisting, but still remains really far from saturation. And so the existence of this intermediate regime is just one way in which the many resource limit is fundamentally different from the simple two resource case that we considered before. Since with two resources, you can only exist either in saturation or in this trivial single species state. Now, with a little work, it's actually possible to work out the structure of this evolutionary steady state in the context of our simple toy model. I've tried to illustrate it in this panel down here, where I sort of projected the resource uptake strategies of the different types in the system on the simple three resource simplex, and the strain frequencies are illustrated here on the y-axis. So what happens is that in this evolutionary steady state, the ecosystem tends to be dominated by a single generalist type, which can actually utilize all the different resources and which is as close as possible to the environmental supply ratio. It'll then be accompanied by a collection of S-1 single loss of function mutations that have recently descended from strategy mutations that occur in the generalist background. And the idea is that the generalist, these loss of function variants are going to be continually driven to extinction by the accumulation of fitness benefits due to pure fitness mutations. They're going to preferentially accumulate in the generalist class by virtue of its much larger size. And this treadmilling of extinction is going to be exactly balanced by the continual production of new loss of function variants that continually rediversify due to new strategy mutations in the generalist background. And so these two effects are going to combine in such a way as to lead to a typical number of species that coexist within the ecosystem at any given time. So it's worth pointing out that while the actual number of species that exists at any one time stays pretty close to this average value S predicted by this formula here, the actual genealogical identities and metabolic resource strategies of these coexisting strains is actually going to be continually turning over in time, sort of a dynamic steady state rather than a static one that we'd expect in an evolutionary context. And this is a really simple example, but I think it illustrates how heritable beneficial mutations can create genealogical correlations within an ecosystem that can drive the ecosystem to this state of diversification-selection balance, which has its own unique set of ecological properties. In this case, we saw that the diversification-selection balance ended up being dominated by some highly atypical ecological configurations that would be really unlikely in the traditional community assembly approach. What would be the odds that we would draw a generalist strain that utilizes all the resources if we assume that the resources are just distributed through some binomial sampling process? Furthermore, at least in the context of the simple model, you can show that the width of this intermediate regime here grows increasingly broad as the number of resources increases. So we might expect these effects to become even more relevant in increasingly large populations. And finally, one of the things that I think is most exciting about this state is that while the number of strains that coexist in the ecosystem at any given time, of course, depends on the number of resources in the system. So that if we pick two ecosystems with different values of R, they'll generally lead to two values of S. If we actually condition on the number of species that we actually observe within the ecosystem at any given time, if we condition on S, then two ecosystems with different values of R but the same value of S will essentially be indistinguishable based on their current genealogical structure, or the ways in which the different strains uptake the different resources. And this is just one way in which the structure of a primitive microbial community can end up being constrained more by the short-term evolutionary dynamics in the population than by traditional ecological factors. I think this is an effect that could potentially be relevant more generally. Okay, so at this point, you might start to be worried a little bit that the dynamics of this evolutionary steady state end up being completely dominated by the existence of this single generalist type. And when we look across at nature, at least it seems to me, at least, that we rarely see ecosystems that are set up in this almost sort of pathological form. And so this leads to a really reasonable question then, what if the emergence of this general type is actually forbidden biologically, for example, because due to crosstalk between different metabolic pathways or limited space on the surface of the cell from expressing too many importer genes. So we can start to get a handle on this, even in the context of our simple toy model, just by imposing an upper limit, R max, on the number of resources that any individual strain is able to utilize. So we just extrapolated from our previous results from before. You might imagine that all that would happen in this scenario is that the resource space would just separate out into different sectors, each of which contains R max resources and otherwise aren't really talking to each other. Within a given sector, we could have then a generalist strain, which is as close as possible to that corner of the environmental ratio space. And again, a collection of S-1 loss of function variants that recently descended from the generalist phenotype in that corner of resource space. This seems pretty reasonable, but it turns out if you analyze this model a little further, you can show that this is actually not what happens at all. Instead, something much more interesting happens. So across a relatively broad range of parameters, you can show that the ecosystem instead remains relatively close to ecological saturation, where the number of coexisting strains is roughly equal to R. And actually, the metabolic preferences of these strains are actually interleaved in this really complex way, which I actually haven't found a great way of plotting on a slide like this. Now, we currently have no idea what the emergent genealogies look like within the population in this case. Well, this is something we're looking into in future work. So I raise this example only to show that the sort of structure of this diversification-selection balance is still a really open question. Okay, so to summarize what I've talked about today, I hope I've convinced you that molecular data suggests that ecological diversification can be a really common and important factor in rapidly evolving microbial populations, and that new population genetic models are going to be required if we want to fully account for these effects in current data. By analyzing one particular model based on competition for substitutable resources, I showed how the accumulation of many linked weakly beneficial mutations can actually dramatically influence the structure of these primitive communities. And they raised the question of whether some natural communities might be poised at this dynamic steady state of diversification-selection balance. We also showed how these acquired fitness differences can then in turn shape the adaptive landscape itself, generating an emergent selection pressure to divert metabolic effort from high fitness to low fitness resources in a way that can actually be dynamic over time as additional mutations accumulate. And now there's a ton of open questions, even in the context of this simple model. But I think one that's really important is an existential one, which is that we've analyzed a really simple model here, which surely can't apply in its exact form in any realistic setting, including even these experiments that we motivated the model with. So an interesting question then is whether, in the same way that we've come to expect with the Wright-Fisher model in population genetics, does this simple toy model actually capture the limiting behavior of some broader universality class, and in doing so, maybe can actually approach some of the signals that we see in data as well? And so this is a much longer term question, but I think with analytical characterizations of the form that I described today, we start to have the tools to be able to tackle this question. All right, so with that, I'd just like to thank my wonderful collaborators on these projects, in particular, Mike McDonald, who's my co-author on the LTE population sequencing project, and as well as Stephen Martis and Oscar Halachek, who are my co-authors on this theory project looking at population genetics in resource competition models. And I just like to thank everybody for their attention, and I'm happy to take any questions. Thanks.
Very nice. That was very slick.
Thanks.
Well-explained talk. I actually have to run in not too long, but a few questions. And of course, like if anybody else has any questions, please tweet or type them into the YouTube chat box. So at the very beginning, you sort of You said that quantitative analysis sort of clearly shows that there's no way you could have these sorts of long-term coexistences without having something more than just sort of fitness that they're just sort of they're not it can't be easily explained by individual fitness models. You need some ecological models.
Yeah.
And so does that just follow from, if you have fitness differences, the time to fixation has to be shorter than the time that you observe this coexistence to be occurring?
Yeah, that's a good question. It's sort of a subtle point, and it sort of dates back to maybe some of these genealogical structures that I talked about at the very beginning of the talk. And so let me just go to where I was describing this in the paper.
Yeah, exactly.
Right. So you're referring to this point here. And so yeah, there's sort of two things going on here. So in a model with clonal interference, you can show that there's a sort of typical time scale for mutations to accumulate within these two individual clades. And there's also a typical time scale for the clade itself to take over the population. And actually, when you work out these clonal interference models, they actually never really look like the simple cartoon I've drawn here. And that's because the sort of time to take over within an individual clade actually can't be too far from the clade taking over entirely, right? So like this mutation here, it goes from a single individual to making up 50% of the population on this amount of time here. The time it takes to go from 50% to 100% should actually be relatively similar. And so this actually shows up if you go back to the genealogies that, let's say, Richard talked about in his phyloseminar. You'll notice it's actually hard to get really balanced deep phylogenies in these simple clonal interference models. There's this one skewed branch here, so this one singleton individual gets traced back all the way to the root here. But actually, this would show up as sort of one clade that exists at 99% frequency and another one that exists maybe at 1% frequency.
Right.
So it's actually really hard to get in these models to get deep branches that are then spindly on the tips that are roughly equal size. And so it's sort of a more subtle point. And I think it shows why it was really important for a lot of work to be devoted towards really figuring out the consequences of these what seem like really, really simple models of asexual genomes, because they allow us to conclude that there could be some interesting behavior going on from data, even without going in and doing the sort of competition experiments where we mix the two types at different frequencies. So those experiments are currently actually ongoing in the case of the Lenski lines. But you can imagine many scenarios, for example, bacteria in the gut microbiome, where we're just never really going to be able to do that convincingly. And so I think it's important to have some theoretical tools to tip us off that something weird's going on.
Right. Cool. So a question from Will DeWitt. So your research strategies are constrained to be in the simplex.
Mm-hmm.
What do things look like if they aren't, so that there's sort of no trade-off in accessing a new resource?
Yeah, okay. So let me just grab the slide real quick. Actually, that's not a good one. How about this one? Yeah, so I sort of motivated the whole talk by thinking about things living on the simplex. This formula here, well, maybe not the formula, but at least the graphical picture here tells you what happens when things are not so constrained. So, the red point here, I've just drawn a perfect trade-off strategy mutation, but you can imagine that a strategy mutation also incurs a cost or a benefit, and we can predict whether it will take over or not, using the same formula that we described before. The really important thing about this simplex is not so much how can new mutations arise that change resource strategies. It's all about these pure fitness mutations. And so if you don't think about trade-offs, if you imagine instead of normalizing things on a simplex, that you just considered the overall uptake rate of resource 1, the overall uptake rate of resource 2, and so on, they don't have to normalize to anything particular, you might think that it would be really rare to get a mutation which just increases all of them simultaneously without shifting the resource uptake rates. But this is the thing that we know is actually happening a lot in these microbial evolution experiments. And so for me, shifting onto the simplex space is just a way of really bringing out the fact that pure fitness mutations could be relevant, and they really force us to grapple with it at the very beginning. A really open question at the moment, I would say, even in these simple experimental settings. Let me see. We have a slide at the end. Right. what actually is the relevant space of mutations that can change the resource uptake rates in different ways. And we have started to have a pretty good understanding of how mutations can change fitness by looking at genome-wide surveys and summarizing things as a distribution of fitness effects. But we really have no idea, even in something simple like the Lenski lines, what is the spectrum of ways in which mutations can change these resource uptake strategies? So I think a big open question going forward is, can we figure out ways of trying to measure this experimentally? And how can we sort of summarize this in sort of an eco-evolutionary DFE, if you'll excuse the buzzwords, that can tell us a little bit about both the ecological structure within the population and how often it's likely to get ecologically relevant mutations that really disrupt the ecosystem state entirely.
Cool. One last quick question. Can you just give a few sentence introduction to what replica theory actually is and how it sort of works? It sounds really neat.
Yeah, it's quite cool, and I really recommend if you're interested. Sorry, let me find the right paper. Some of these references down here are some really good introductions to how this works. And so the idea is the following. So we draw a collection of random colonizing strains. So we pick some number S0. This is the number of species that colonize the ecosystem. And then we randomly assign them resource uptake strategies according to some fixed distribution. And so in practice, you typically have to assume that these distributions have some simple form. Things are Gaussian, or maybe they're like a binomial distribution about whether you use or don't use a different resource. And you assume that these are then fixed for all time. Whereas the current frequencies of the strains and the current concentrations of the resource are sort of internal degrees of freedom that are then dictated by this initial random choice. And so essentially replica theory, I don't know, the way I think about it maybe is a way of sort of exchanging these two limits, where you can think about a really large ecosystem and then average over the typical internal state for a fixed draw of these things, and then do a second average across the different random draws of the resource strategies. And so this only works reasonably well when you have this sort of self-averaging behavior take place, where you end up having a lot of random things contribute in a way that starts to cancel out a little bit from the central limit theorem. And in these cases, you can show that it doesn't really matter so much what you pick for these initial random resource strategies and fitnesses. They all, because of the central limit theorem, they essentially all tend towards Gaussian random variables. And so replica theory is all about figuring out what's the effective mean and the effective variance of these sums across the different resources, and in a way that's sort of self-consistent at equilibrium. So I'm not sure if I've really done it justice there, but if you're sort of interested more, I'd really recommend reading some of these papers. It's a really cool approach that has been really useful in ecological contexts. And there may be some role for applying this in evolutionary settings as well. You just have to be a little bit more careful with the correlations between the different strategies in that case.
Right on. Okay, perfect. Thank you very much. I do have to run now. So that concludes our series. Thanks for doing such a great job finishing it off. And we'll see you folks in the fall.
Awesome. Thanks very much. Thanks, everybody.
Bye.